Well, hello to everyone, and thank you for joining us today. This is Sam George. I'm Senior Medical Advisor to New Cardio, and today we're going to talk about fully automated approaches to assess drug cardiac safety during early clinical development. Now, there should be in the webinar then an opportunity to ask questions via the chat window. In general, I'm going to save the answers till the end. Just go ahead with the presentation and save hopefully about 15 minutes at the end to deal with questions and discussion. So with that, let's get started. So what are we going to talk about today? To just give you a quick overview, I'd like to talk about the current status of cardiac safety evaluation in early phase studies, particularly the current approaches to ECG evaluation for cardiac safety in early phase. The emphasis here is going to be on the critical role of measurement accuracy and precision in ECG analysis and how fully automated approaches deal with that. I want to talk about the potential advantages of fully automated approaches over current manual or semi-automated approaches. And I'm going to emphasize here QTinno, because that's what I have the most experience and knowledge about, the QTinno fully automated software for ECG analysis. And I'll tell you about its key technical features that assure both accuracy and precision. And then we'll go over the results of the clinical validation studies that we've conducted so far. Subsequently, then, hopefully we'll have time for questions and discussion. So that's kind of a quick overview. So let's start with, kind of the current status. Clearly, the E14 guidance and its updates have had a tremendous influence on cardiac safety evaluation. They've set very exacting standards for cardiac safety evaluation. And what E14 states and what has become the norm is that all new drugs have to have a comprehensive ECG evaluation early in development to determine their effects on primarily cardiac repolarization and to attempt to identify those drugs that may have a arrhythmia risk associated with them. A key feature of the E14 is the thorough QT study, which is a dedicated placebo-controlled blinded trial to determine whether the drug induces QT prolongation that exceeds the threshold level of regulatory concern. It includes a positive control arm, which is usually moxifloxacin, to assure that the study is sensitive enough to detect QT prolongation. And moxi is a known modest QT prolonger. And the standard set forth in the E14 for a positive TQT study, or the existence of a QT signal, is 5 milliseconds. But what they're really looking for is a 95% upper confidence limit on the double delta, that is the placebo and baseline controlled change in QT, that exceeds 10 milliseconds at any of the time points in the study. So it's a very exacting standard that's set forth and that sponsors have to meet. It's not just the thorough QT, though. There are equal demands, or very important demands, for accuracy and precision in any early phase study, MAD-SAD study, so forth. Well, what happens if you do exceed that threshold level of regulatory concern? You are now have a positive thorough QT study, and what the E14 states and what happens is you have to enter into an expanded ECG safety evaluation in phase 3, later stages of clinical development. And that's going to vary with the drug and the nature of the QT signal, so forth, but clearly there's going to be additional demands in later phase development if you have a positive thorough QT, and that can add quite a bit to the cost of drug development, maybe quite a bit to the time as well. So much so that it could affect a positive TQT signal, could affect a go, no-go decision for further development in some cases. So it's a very significant event. Okay, what at a 40,000-foot level are we talking about? What does a thorough QT look like? And we're looking here at the hourly change in placebo-controlled baseline subtracted drug-induced QT changes, the double delta QTC corrected QT. And this would be a typical example. This is not actual data. This is, kind of hypothetical data of what a modest QT prolonging drug would look like in a thorough QT study. You get prolongation in this hypothetical example of about 7 milliseconds, a little bit of QT prolongation, but the upper confidence interval on that, when it gets up to about 7 milliseconds, is less than 10. It's under the threshold level of regulatory concern for the 95% upper confidence limit of 10 milliseconds. So it's a negative study. But you have to have another piece of data to show a negative study, and that's the positive control. In this case, MOXIe, which is shown here. So in a crossover or parallel group study, moxifloxacin is given, and the double delta on that, there should be a signal. It's expected to prolong the QT. And this is actual data from a thorough QT study. And it shows a typical rise and fall of the MOXIe signal, reaching a peak of about 3 hours, 4 hours. That's about 11.5 milliseconds. And then it gradually declines to about half the level by 24 hours. So that's what a MOXIe curve looks like, and that's what the FDA expects to see. Now, a key feature of this is that at least one time point, the 95% lower confidence limit has to exclude 5 milliseconds. If none of those time points excluded 5 milliseconds, you would have a failure to detect the MOXIe signal. You'd have a positive control that didn't show the expected signal, and you might get sent back to redo the study in that circumstance. But here we have multiple time points that exclude 5 milliseconds. So this is an example of a negative QT study. The drug did not exceed the threshold level of 10 milliseconds, and the MOXIe signal is present and clean. All right, so looks simple, but it isn't. There are some real issues. The measured QT interval is highly variable, and that makes it difficult and technically demanding to detect a little bit of QT prolongation. What's the cause of the variability? First of all, there's normal physiologic variability in everybody's QT interval. It varies inversely, first of all, most importantly, with the heart rate. That is, as it accelerates, the time from the onset of depolarization to the end of repolarization, the QT interval, will decrease. And when it decelerates, it'll increase. So it's very much affected by the heart rate. And so you definitely, for the phase 1 unit, controlling the heart rate is very important. You have to have the patient supine, resting, comfortable during the designated time period. QT intervals are also affected by the autonomic state of the study subject, and this goes beyond the autonomic state's effect on heart rates. It directly affects repolarization. So it's important to control that, again, with the subject being supine and resting. You don't want to give meals during the treatment period. You don't want to do a blood draw during that designated time period. You don't want to affect the autonomic state of the patient. Finally, QT intervals are affected by what's called hysteresis. That is, if there's a sudden change in heart rate, it takes a while for the QT interval to catch up to that change in heart rate, either acceleration or deceleration. And so it's very important to have a nice, stable period of stable heart rates. If the heart rate changes, it can take a couple, 3 minutes for the QT interval to catch up, and that can induce variability. There are also technical and measurement-based causes of QT interval variability. First of all, the quality of the ECG recording. Obviously, if you have a noisy recording, it's harder to read, it's harder to determine QT interval, and that's going to induce variability. For manual and semi-automated approaches, there's inter and intra-reader variability. Two readers are going to pick different endpoints or pick different fiducial points just because of individual variability. In fact, if you show the same ECG to the same reader 10 days later, he or she is going to have some different measurements. There's intra-reader variability. Finally, it can be affected by the ECG lead that's selected for measurement. Most manual semi-automated techniques are single lead, and you want to be very, very consistent about reading from the same lead. On the right, you can see an example from the same ECG where if the QT interval was determined from lead 2, it would be much shorter than it would be if the reader measured the QT interval on V5, and it's quite a significant difference. So the lead can very much affect, the lead chosen can very much affect the QT interval, and you want to be very consistent about that. And this is going to come back. We'll see how this affects the overall study. This kind of variability will affect the overall study. Now, if you want to drill down on this, I highly recommend Jean-Philippe Couderc's review in Journal of Electrocardiology journal last year, volume 42, 494. That's an excellent review with lots of great references that gives a great overview of the whole topic. And if you want to drill down, I highly recommend that to you. Okay, what's the effect of QT variability? It very much affects the overall study, and it can affect the number of subjects needed. It can affect the study power. What are the factors that affect the number of subjects needed or study power? First of all, the significance cutoff. Well, you have no control over that. That's set by the FDA at 95% or P of 0.05. So that's one of the things that affects it. The other thing is the maximum allowable QTc effect, and that's 5 milliseconds. It's mu2 minus mu1 minus mu2, and that's 10 minus 5 or 5 milliseconds, and that's also set by the FDA as the threshold of regulatory concern. The next thing that affects study sample size is study power, and that's entirely within the sponsor's discretion. It can be set at 80 or 90%. Whatever the sponsor feels like, but the higher the power, the more subjects needed. But the last thing that affects the number of subjects needed is the variance, or the variability of QTc, the variance being S squared. And that is something that you really want to strive to control, first of all, with good quality subject control and ECG collection, controlling subject variability at the phase 1 unit as scrupulously controlling that as best you can. And then finally, measurement precision. You want to highly precise, consistent, non-variable way of measuring the ECG. All right, so what are the current ECG measurement approaches? One approach is manual, and that's where cardiologist or technician puts the fiducial points on a digital ECG that's displayed on a high-resolution monitor. Usually reads 3 consecutive complexes from a single lead, most often lead 2. And so for triplicate ECGs, typically a total of 9 beats are measured for each subject and each time point. Obviously, that's the most labor-intensive of all the measurement options. Another approach is semi-automated. It's similar to manual, but this is where the initial fiducial point placement is made by computer algorithm, again, most commonly on a single lead with the computer algorithm determining T-end by the tangent baseline intercept methods. There are other methods for determining T-end, but that's the most common. Then after the initial fiducial points are placed, then all the ECGs are reviewed by a cardiologist or a trained technician, and if adjustments are needed, they're made at that. And again, it's typically 3 consecutive beats, a total of 9 beats for triplicates. That's a little less labor-intensive than manual, but it has a certain disadvantage, and that's that humans are going to read a little bit differently than computer algorithms. When you get in and start adjusting them, you wind up with a mix of computer and human measurements, and therefore two different kind of approaches to reading the ECGs placing fiducials, and you wind up with a mixed data set, which can potentially increase the variability of the results. Now, the next method is now fully automated. There's a kind of a gray area between semi-automated and fully automated, but the general characteristics of a fully automated measurement method would be that the computer measures all the intervals and also makes an automated quality assessment so that it can flag a small percentage of the ECGs where there may be noise issues or heart rate changes or so forth, and culls them out for human review so that a human can look at them and determine whether they're acceptable or problematic reads. The advantages of this, obviously it's going to accelerate the study a lot. The QTinno method can fully analyze 10 to 20,000 ECGs per hour of CPU time, basically on an ordinary desktop, which is going to reduce study costs. Obviously, it's going to eliminate reader variability and other sources of measurement variability. It's very precise and completely reproducible, and requires either no human intervention or a bare minimum, at least, to ensure that you don't wind up with mixed results. The human will look at the problematic ECGs that are culled out, but in general, no adjustments are made. It's fully automated. So let's review the new cardio solution, which we call QTinno. There are others that are, I think, very good, but this is where I have data and experience and can discuss in an informed way. So QTinno is new cardio's fully automated solution, which determines all cardiac interval measurements and also assesses T-wave morphology in an automated way. So all measurements are made on what we call the heart vector magnitude lead, which is a virtual lead that I'm going to discuss in either the next slide or one or two slides ahead here. It's a very noise-resistant approach that includes all of 12 leads. Synthesized down into a single lead, so that you get global cardiac information. And we think that's an advantage. It analyzes all of the complexes in the ECG, and the more complexes you read beyond 3, the more you can reduce variability. And it analyzes all of the complete complexes in each ECG. It has intelligent automation with the self-validation that ensures accuracy and reliability of the measurements. And it has what we call confidence factors, which I'll discuss in the next slide, which accurately identifies and flags these problematic ECGs, noisy, some issue with them, their intervals outside of a expected range. It will identify those and flag them for human review. And finally, it provides the ability to overview any of the ECGs in there, and you can make manual or adjustments to the read if you wish. We don't necessarily recommend it, but you could. A manual reader could override. The inputs, QTinno will accept standard ECG files from any ECG equipment, and there's no need to change any of the collection practices. It takes standard digital ECG input. And that initially performs an assessment of ECG quality, identifies seriously defective ECGs like severe baseline wonder, high frequency noise, missing leads. So it's usually a fraction of a % of the ECGs that have these real serious issues, and it identifies those right up front. And then it goes on to identify the problematic ones for overread. All right, what are the key features of this? I talked about this vector magnitude virtual lead. It takes all the 12 leads, information from all of them, it transforms it into a three-dimensional vector, and then it calculates at each time point the magnitude of that vector in all 3 planes. The bottom line is you wind up with something that looks like an ECG lead, but it's actually a virtual or calculated ECG lead. So you have the full information. You have from the very beginning of the Q to the very end of the T, so you don't have this variation in 12 leads, a difference between the two. You've got all the information in a single lead, and that improves the signal-to-noise ratio, and we think is more reliable and more consistent way to measure the QT interval. Now, a second feature that ensures accuracy is some iterative curve fitting algorithms that optimize the placement of the fiducial points. And the key feature of this is the use of a least square fitting of a third-order polynomial to the vector magnitude data, and then it does multiple iterations to find the best fit where the minimum of that function fitted to the raw data represents the t-end, or the fiducial point that you're seeing. That's a very noise-resistant approach. It's also independent of baseline. You don't have to know what the baseline is, and sometimes the t-end is above or below the baseline. That can confuse tangent intercept methods. But this is baseline independent, and that's very important, we think, for accuracy. All right, confidence factor. I've discussed that. If you're going to fully automated read, there's got to be an internal assessment of the quality and reliability of the reading. Otherwise, you're stuck with two very undesirable options. You've got to accept all of the automated results on a black box basis, having never looked at them, or if you haven't done this confidence factor or similar analysis, you've got to overread all results to make sure that there isn't some problem there. So the confidence factor assessment that culls out the problematic ECGs is critical. Otherwise, you're really stuck with a couple of bad options. All right, so what does confidence factor do? We arbitrarily have a scale of 100. We assign each ECG a confidence factor of 0 to 100 based on the signal quality, excessive noise or missing leads, etc., but also on the measurement quality, and that's the goodness of fit of that third-order polynomial. How well does it fit? It also takes into account things like heart rate variability, all the factors that you would want to look at if there's an issue. It identifies the ones where the QT interval is out of range, and we'll flag those, so forth. And we currently have set a confidence factor of 70 as flagging for possible overread. And that will typically represent about 2 to 4% of a phase 1 or thorough QT study data set, a small percentage of those. And by overread, we just mean take a look at them, not to adjust them. And the overwhelming majority of that 2 to 4% are perfectly adequately and correctly read, but to take a look, there's an issue. How well does it work? This is basically a comparison of the QTinno and Core Lab by CF category, and the standard deviation of the difference between the two in 3 of the studies that we've done. And you can see when the CF's above 0, when the confidence factor is above 0, the standard deviation of the difference is very tight. There's always some difference, but it's very tight. But it increases when the CF is 0, when you've got a noisy or difficult ECG. That doesn't say that either QTinno or the Core Lab read is wrong. It just means that that's a difficult ECG, and there's obviously, therefore, more variability between the two. And typically in a phase 1 study, the confidence factor of 0 is going to be less than 0.5%. The one that I just finished today was under 0.3%, so a very small number. All right, so when processing is completed, QTinno is going to provide an ability to view all the processed results, gives you all the confidence factor results. If any adjustments have been made by a manual reader, it will note that so that you'll have information on if any adjustments have been made. Provide the study data results in a common delimited file, which can be imported into any standard statistical program. Provides the annotated XML files for submission, and of course then all the additional study reports and business aspects of the, that you need. All right, so let's get into how have we validated QTinno. What have we done? How has it been validated? So we have several studies. 6 of them are shown here. We've done, I think, at least 2 or 3 more since this slide was made. And this gives you a 40,000-foot view of the clinical study results. Now, NCE 1 through 3 were all multiple ascending dose studies with NCE 1 and 2 were quinolone antibiotics, NCE 3 was sotalol. And you can see in those 3 studies that the core lab and QTinno were highly concordant. The difference in the mean delta QCCF and double delta was less than a millisecond in all cases. And the ECGs flagged for overread generally range from about 2 to 5%. In the NCE3 study, which was a high noise data set, which was extracted at 180 hertz from Holters, almost 10% were identified, but that was a very difficult data set. We've also analyzed retrospectively, I think now 4 or 5 TheraQT studies. 3 of them are shown here. Again, highly concordant, with the little exception of TQT2, where the core lab was reporting more outliers than QTinno, and we'll get into that a little bit. But the difference in mean delta QTC, again, and the number of ECGs flagged for overread were very low. And you can see the CPU time, of course, was very, very fast on all these studies. So there's a tremendous time advantage and human effort advantage for fully automated reads. We can't review all those studies here, but we can review 2 as examples. And I'm going to compare for you the QTinno validation results on 2 of the TheraQT studies, what we call TQT1 and TQT2. I'm going to review these because it's kind of instructive. They're 2 different design studies, and the results that we get, I think, are instructive. So the TQT1 was a parallel group study, about 11,000 ECGs. And what we call TQT2, we obtained from the CSRC, the Cardiac Safety Research Consortium. And that was a crossover trial of about 18,000 or 17,500 ECGs. And both of these were only the Moxie and placebo arms that we were given. Now, TQT1, it was fully blinded, and the analysis was done by the pharma sponsor. For TQT2, we were provided that in an unblinded way by the CSRC. We did the analysis ourselves. So what do we get? Let's just look at the Moxie curve. For the TQT1 study, the parallel group study, here you can see for both core lab and QTinno, both show an excellent Moxie curve. Typical Moxie signal with a rise and fall, peak here at 4 hours, then declining. Multiple point estimates where the 90% lower confidence limit excludes 5 milliseconds. So it's detected the Moxie signal in both instances. And the 90% confidence intervals are very comparable here. Slightly narrower for QTinno, but very close. Now, the TQT study, the crossover study, again, you see a very typical Moxie curve, rise and fall, multiple time points, excluding 5 milliseconds. Very little difference, as was the case in TQT1, in the point estimates, less than a millisecond difference at 7 of the 12 time points, and the confidence intervals overlapping between the 2 methods at every time point. So there's no statistically significant difference at any time point. But here, in TQT2, the 90% confidence intervals for QTinno were consistently about 50% narrower than the core lab. Now, the core lab's very narrow. It's got a great Moxie curve, but the confidence intervals for QTinno are about 50% narrower at every time point. And so that is an indication that there is less variability in the QTinno readings than in the core lab, and that could be an advantage. All right, so why did we see that rather sizable reduction in confidence intervals? So we looked at these, and we looked at them very carefully. Now, these are Bland-Altman plots, and Bland-Altman is a nice way of comparing results from two different measurement methods, and we're comparing here the delta, or the change, the baseline corrected change in QT between the two methods, what they came up with. And so you can see on the left half here, there's some where the, on the x-axis here is the average reading between the two methods. And, you know, some of them had shortening, some of them had prolongation. There's an overall very good agreement, though. The mean difference is 0.1 milliseconds. The standard deviation was 8.1. So there's a pretty tight correlation between QLab and QTinno for the results here. Now, TQT2, again, we see a very low mean difference. Essentially, the mean difference is 0. The standard deviation, again, there's not a broad difference, but there's clearly a trend here, a downward left to right trend, something we've never seen in any study. And so we were surprised by that. And what is going on here is that in the upper left-hand quadrant, QTinno is reporting a less negative delta QTC than the QLab. And in the lower right-hand quadrant, QTinno is reporting results that's less positive than the QLab. And why would that be? Again, we'd never seen this before. So the only way to figure that out that we could figure was to actually look at all of the outliers, all the ECGs and all the outliers. Now, each one of those data points represents 3 baseline ECGs and 3 either on drug or on placebo ECGs. So you've got to look at, there were about 30 outliers in each direction. 60 times 6 is 360 ECGs you have to look at and ask yourself, why is there a difference? Because we were unblinded here, we had the good fortune to be able to have the actual QLab data. So we could look and say, okay, on this ECG, the QLab read this, QTinno read this, what's going on? So this is what we found. In 80% of the cases where there was this, an outlier in either direction, we observed the following, that some of the ECGs were read looks a little bit strangely. Now, when you look at the QTinno TN, the red line, it looks like a good placement. When you look at the QLab TN in these outliers, it looks early. It looks, if you look at V5, it doesn't look like TN at all, unless you look at V2. And on V2, it looks like it's exactly right. And so in these outlier ECGs, we think that in an inconsistent way, somebody was reading V2 in those issues and creating some variability. So how does that happen? How does that cause it? Well, the impact of this V2 issue, as we call it, in the upper left, it looked like the on-drug ECGs were being read on V2, but not the baseline. And that caused the QLab to be reported as more negative than QTinno, because the on-drugs were being read on a shorter lead. In the lower right-hand quadrant, it affected the baseline ECGs, but not the on-drug. And that caused the delta to be reported more positive. Well, in the end, that doesn't affect point estimate. That always averages out. So the point estimates that you saw, they were the same. But it does affect variability. It increases variability, and that's to be avoided. If you add variability, you're going to either reduce study power or cause more, a problem with respect to power, to number of subjects. A more recent study that we've done is a Holter-based study on QT4. We call it, that's a fully blinded analysis of MOXIE and placebo from a crossover trial. This was entirely Holter-based. It's the largest study we've done to date. We had 2 24-hour Holter recordings on 71 subjects. We had to carry a hard drive to the sponsor. We couldn't get it on DVDs. We actually had to carry a 250 gig hard drive to get all the data. We examined a range of automated parameters that we could adjust and play with to further improve the QTinno results and further reduce variability. Again, as for the non-Holter-based studies, an excellent MOXIE curve, less than a millisecond difference between the core lab at virtually all of the time points. The 90% confidence intervals were narrower at all the time points by an average of about 20%. And some of the interesting things we observed in this Holter-based study were, in general, the larger the window that you extracted the data from, the more the variability decreased. And if you read all the good beats in that entire window, 400, 500 beats, that actually reduced variability as well. So there's a real opportunity, I think, for the future in Holter-based studies to further improve the quality of the data, and we're very excited about that. All right, let's take a quick look at the variability in all the studies that we've done, comparing the residual variability of core lab to QTinno. And remember, that's important because that's the S in that number of subjects needed. So the lower the number, the better, the less variability, either the more power you have in the study or the less subjects that you need. And this was done with a mixed statistical model on SAS with factors for period, age, gender, treatment, all interactions. And you can see here that QTinno in every study had a lower residual variability than the core lab, sometimes a little bit as an NCE3, and sometimes quite a bit as an NCE1 and TQT2. So that's important, reduce variability, and I seem to have paused here. Are we seeing the screen? I hope so. I'm having a little bit of a technical issue. Let me try this again. Okay. Okay. So again, just to end with this, why is measurement precision important? A recently published study, I think, really drives home the point. And this was a thorough QT study of a alpha blocker for BPH, a typical parallel group study with 4 groups, 45 to 48 subjects each, low and high dose active drug, and then a placebo and MOXIE arms. Okay, so let's look at the active drug, the QT signal, and there is none. The drug does not prolong QT. There's no time point where the 95% upper confidence limit, single-sided, exceeds 10 milliseconds. So it appears to be a negative signal. The drug doesn't have any QT prolonging effect. But what happened in the MOXIE arm? Well, here is the MOXIE data. Oops. And we can see that the MOXIE signal is weak. And in fact, there is no time point where the 95% lower confidence limit exceeds 5 milliseconds as required by the ICH E14 document. So in essence, that's no MOXIE signal. There is some rise of some prolongation that's being caused by MOXIE, but the 95% lower confidence limit does not exclude 5 milliseconds at any time point. So why is that? It looks to us as though this was imprecision, that there were very wide confidence intervals and a very large standard deviation, which we haven't observed before. Now, that, whatever the cause is, I don't know, but that's what caused those confidence intervals to be large and to fail to exclude 5 milliseconds. The previous data was showing no MOXIE signal, or no active drug signal, but there's no MOXIE signal either, as would have been expected. And so you could get sent back to do a whole new thorough QT study if this happens, and that drives home the point you've got to have good precision. There's less risk, first of all, of a false positive study, that is a drug that really ought not to have a QT signal, but the 90% upper confidence limit fails to exclude 10 milliseconds. There's less risk of a failed thorough QT study due to an insufficient MOXIE effect, and you can do an adequately powered study with fewer subjects. All right, let's summarize. the QTinno fully automated analysis is highly accurate. We got interval duration measurements and double delta MOXIe curves that were closely comparable to the careful manual or semi-automated adjudications in all clinical validation studies today. All of the QTinno data I showed you, let me emphasize, was fully automated. There were no human adjustments in that data that I showed you of any kind. It's fully automated data. Yes, we looked at the 2 to 4% that were flagged, but we made no changes in the data. Okay, so it's precise and reproducible. The variability of the QTinno measurements have been lower than other techniques in all of the validation studies we've done to date. And we've discussed the impact of that lower variability on giving a higher study power the opportunity to maintain the same power with fewer subjects. All right, what are the current efforts we're making to further improve? I think I mentioned the Holter-based studies. There's a tremendous opportunity there, I think, to further improve cardiac safety assessment. I think there's a real advantage to doing Holter-based studies over the standard 3 10-second ECG approach. We're also looking at newer biomarkers that may improve accuracy of the QT assessment, and it also may provide the ability to detect drugs with direct cardiac toxicity, for example, cancer chemo drugs. So we're very enthusiastic about that as well. To just end, I want to point out that we have now a partnership with Clinilabs that we entered into recently, and studies can be conducted at the Clinilabs CRU with either ECGs or Holters, and then analyzed by QTinno. It's available for any QT study and early phase studies or thorough QT study, and there's the contact information if you'd like to know more. And let me end with that and see if I get questions. It's open for questions, and we'll wait a little while for them to come in, and I thank you for your attention and your time. While we're waiting on questions, I want to mention, there are other very fine, highly automated approaches that I don't know a lot about, and I didn't show their data, but I certainly meant in not showing that, no disrespect. The data that I've seen in presentations shows good MOXIe curves, good variability, so forth, and so there are other approaches. Are there any comparisons between the two? No, there are no direct comparisons. The CSRC has now made available a blinded data set that they're going to use to evaluate fully automated approaches. They are very scrupulous about saying we're not going to make any value judgments or direct comparisons. We're just going to run the study in a blinded manner and post the results. And so that's a very good way to, I think, to further validate the study, and we're participating in that and look forward to seeing others participate in that as well. No questions yet. Must have been very convincing. Okay. Got one coming in here. This is a very good one. What will it take to have the FDA or other regulatory agencies accept automated studies for thorough QT studies? Well, they have been very clear that they are more than open to accepting fully automated results if they're validated. And the validation that they're looking for is, can you accurately detect a MOXIE signal in the database or the data being submitted? Can you accurately detect the positive control? We visited them in August of 2009, the IRT for QT. We made a presentation to them. We showed them our data, and they said, yes, this is appears to be validated, and if anybody has any questions or concerns about submitting data with this approach, they can just contact us, send us an email, so forth, and we'll let them know. So the FDA is more than willing to accept the standards that they would be looking for would be the ability to solidly, accurately, and precisely detect a MOXIE signal, and we've done that. All right, so another question. Since fully automated analysis seemed to result in lower variability, do you assume this lower variability and thus enroll fewer subjects for the same amount of power? Well, that's going to be up to you. That's one of the options. The answer is, if you reduce that S, at least in theory, you could reduce the number of subjects. Now, that's a very important decision. These are critically important studies for sponsors. And so the question would be, on the table would be, well, do you want higher power, less risk of a beta error, or do you want to reduce the amount of resources required, reduce the number of subjects? And that's a discretionary decision that remains with the sponsor. But theoretically, at least, you could reduce the number of subjects and have the same amount of power. Any downsides that you can see to the automated methods? Can things be missed? Another good question. Well, I think the downside is, well, what if you had inaccurate reads in some of the ECGs and they weren't identified? And you weren't aware of that. So you've got to have a reliable method, automated method, for identifying those ECGs. And we think the confidence factor method, after having reviewed 80,000 ECGs, we think the confidence factor method is extremely powerful. And can you say there's no chance? Well, we've never seen it, but if it's identified as high confidence, which is 96% of the ECGs in a phase 1 or third QT study, 98%, it's identified with high confidence. We've never seen an inaccurate read. If it's CF0, that's where we see problems. So the key thing is an accurate method for identifying those that would assure that you don't miss inaccurate reads, and we think we have that. Can doing, good question, earlier SAD-MAD studies with automated ECG collection replace the thorough QT or give an earlier view? Well, that's a good question too. And I don't think that it could replace the thorough QT. The SAD-MAD studies don't typically include a MOXIE arm, and so the FDA at least is very insistent that you've got to have a positive control group, and they wouldn't have that. But can it give an earlier view of cardiac safety? Absolutely. I think if you can look at the SAD-MAD studies, and they're not powered for looking at QT, but because you've reduced the variability, you've increased the power of those SAD-MAD studies, you can get a very good sense, I think, of what the likely signal is going to be. Is it likely to have an issue? Is it likely not to have an issue? You can get that from a SAD-MAD study, and I think, therefore, fully automated approaches such as particularly QTinno, I think are ideally suited to giving that earlier view of cardiac safety, and I think it's ideal for them. I think it's also ideal for PK/PD determination. I think we've done a few of those, and you get a very, very nice, accurate, thorough view of PK/PD correlations with that. So in both of those instances, I think the automated ECG analysis is going to have a significant advantage I don't think we'll replace the thorough QT study with it because SAD/MAD studies aren't designed with a positive control, so forth. But I think you'll get an excellent earlier view of cardiac safety. And I don't see any additional questions. I'll leave it open for questions for another few minutes, and then if there are no more, we'll end the presentation. But I'll leave it open for a minute here. Well, since there seems to be no more questions, why don't we go ahead and wrap up? I very much thank you for your attention and your attendance. The questions were superb, and I look forward to getting together with you all at some point. And again, appreciate your time and your interest. With that, I'll turn it back over to Javier, and we'll end.